lecture up online? Because it is going to be a Friday, and most people skip on Friday. Is there any way I could email you and get a copy of it?
I haven't done that yet, so I can't say if I would or wouldn't, but yeah. Okay. I'm not planning to not put it up.
Okay.
Yeah.
I just wanted to make sure I didn't.
Okay. Well, thanks for letting me know.
So the finals tomorrow in this room?
The final?
I mean, midterm, I would say.
Yes.
In this room this time, right?
Yes, sir. Good answer to the question, by the way.
Yeah. So how exactly is glucose avoidance? Just really reactive if it's not been phosphorylated?
So high concentration of glucose will cause cells to swell and burst. It's osmotic. It causes osmotic pressure change.
And that's why cells shut off. So say if you get a high dose of like refined sugars, your immune system shuts down.
That's a different phenomenon. But if you think about it, you can leave jelly sitting out on the counter for a long time before anything happens. The only thing that'll ever attack it is mold. You won't see bacteria get it because high concentrations of glucose kill them. Yeah. Cool, huh?
And is it just because it floods into the cell because of a low concentration?
Cells can't grow in high concentrations of glucose. They can't do it.
But isn't it the glucose entered the cell? Isn't that regulated by the membrane?
Right. So you have osmotic pressures. Right. Okay.
And the osmotic pressure just overcoming the proteins.
Cell will burst. Yeah. No, I'm always there by 10. I'm always there by 9, actually. But I'm trying to think if I've got.
I have like I did last time, just a bunch of last minute.
That's fine. Let me see.
Clarifying questions. Or is there
Tomorrow.
a specific time you want me to come back?
No, I'll be there. Okay.
Perfect. Thank you.
Okay, folks. Let's get started again. Yes, ma'am.
Peptidyl Transferase, is that in the large subunit?
Okay. Peptidyl transferase is that in the large subunit. So peptidyl transferase is the description of an activity, and that's the activity that makes the peptide bond. And that activity is part of what I called a ribozyme, meaning that it's part of the 23S ribosomal RNA, and that is found in the large subunit, yes.
So it is part of the ribosome?
So it is the 23S RNA acts as a ribozyme. Don't confuse ribozyme with ribosome.
Right.
Right? So 23S acts as a ribozyme, yes.
And it controls the peptidyl transferase?
It is the peptidyl transferase. So the peptidyl transferase is simply an activity, and the activity comes from the fact that it's a ribozyme. Let me just check my camera. Go ahead. Carry on, Kevin. Yeah, okay. I was afraid I hadn't turned it back on, but I did. All right. Does that make sense? Yes? No? All the above? No? Okay. So when I say something's an activity, it's just an observation about a catalysis, right? That activity could come from an enzyme. It could come from a ribozyme, right? Breaking down glycogen is an activity, right? It comes from an enzyme known as glycogen phosphorylase, right? So making peptide bonds is an activity, and the activity comes from the ribozyme known as the 23S ribosomal RNA, right? No? Yes?
I was wondering if you could talk about selenocysteine and the difference between direct incorporation versus post-translational modification.
Oh, good question. Very good question. Selenocysteine. Okay. So selenocysteine is what I described in class as the 21st amino acid. And I noted that it got into proteins not as a result of post-translational modification, but instead it was built into the protein as it was being made by the ribosome. So selenocysteine is at a low frequency attached to a specific tRNA. And that's necessary for it to get incorporated during translation. That's what's happening there. Okay? And that contrasts with what would happen if we did post-translational modification. So post-translational modification, I mean, after the protein was made, we modified an amino acid. So one way we might do it, you could imagine me putting a selenium inside of it. And what I said was that's not what happens. It gets put in as that amino acid, as selenocysteine, and gives the protein the final amino acid there. Does that make sense? Yes, Lisa.
Where does fructose 2,6-bisphosphate come from?
Where does fructose 2,6-bisphosphate come from? Unicorns.
Sweet.
Did we talk about that at all, or it was just,
I'll tell you. We didn't talk about it. And I didn't tell you because it often confuses people. So now I will tell you. You're not responsible for it if I tell you here. Okay, I won't tell you. Well, if I ask you that question on the exam, you can say unicorns. How's that?
So it just is there to regulate.
What's that?
We just talked about it because there's regulation of glycolysis.
It's a molecule that reciprocally regulates glycolysis and gluconeogenesis. Right.
What does it act on again?
Okay, so it's a reciprocal regulator, so it activates PFK, which is part of glycolysis, and it inhibits fructose 1,6-bisphosphatase, which is part of gluconeogenesis. So that's how it reciprocally regulates those two pathways. Yes?
I'm pretty sure I understand the idea,
Yep.
but will you go through number 2 on the problem?
Okay, so Gibbs free energy, did you have a question about a specific one, or maybe just talk about it in general? Number 2. Is this on the ones I've got on my thing? Okay. Let me pull it up since I forget what I've got on there. Okay. And that was there? Oh, there it is. Okay. Number 2. At equilibrium, the ratio of products is 2 to 1. What is the value of delta G 0 prime? Okay. How would you solve that? Sure. Why don't I write it on the board, I guess? No, I'll write it on here. Even better. And let's see. Source and center and... Somebody got a pen? I'll have a pen. Borrow a pen. Okay. It's not coming. Here we go. Okay. All right. So what was it? So let's start with number 1. So I've got 300. Okay. Is that showing up? No. R, I'll leave as R. And I've got A going to B. And at equilibrium. So when I say at equilibrium, what's the first thing that pops into your head? Delta G equals 0. Okay. Delta G equals 0 equals delta G 0 prime plus RT, natural log of products all over reactants, right? Which in this case is B all over A, right? All right. So, and I said the ratio was products to reactants is 2 to 1. So, in order for this to be the case, I would have to say delta G 0 prime equals minus RT times natural log of 2 over 1. Right? If you wrote that on the exam, you'd have the answer for number 1. Right? Then in the next one, it says if the ratio of products to reactants is changed to 1 to 2, will the forward or the reverse reaction be favored, and why? All right? Well, when I change the concentrations, am I going to be at equilibrium? No. Right? Well, how am I going to determine the direction of a reaction? What determines the direction of a reaction?
Whether delta G is negative.
Negative or positive, right? Okay. So What's the value of this log term? Is it positive or is it negative? And what's the value of temperature? Is it positive? And what's the value of R? Is it positive? So this whole term, the value is negative. Right? The minus sign. Right? So the delta G0 prime is minus. All right? So now I've got a new equation. I've got delta G equals a negative number. Right? We agree with that. Okay? Plus RT times the new concentration, natural log of 1 over 2. Right? Is this a positive or a minus? Positive, positive. So what's the value of this? What's a minus plus a minus equal to? I hope it's a negative. Right? So the delta G is negative. The direction of the reaction is forwards. Make sense?
Yeah, but I think the answer is like 3.88 or something. You don't care if we have the exact number.
I asked you the direction, right? Will the forward or the reverse reaction be favored, and why? Right? Make sense? I'm sorry? I can't understand. I can't hear you.
Show the answers.
Oh, solutions here? Okay.
RT times L and 1/2 whatever your answer is.
All right, let's see. Where are we at here? So, okay.
1.61 is the answer of the RT, so I can multiply that by 2 and get 1.22.
I don't have a calculator here to do that number, but I mean, the point is it's a negative number, right? So I said the question was, will it go forwards or backwards? And I said it goes forwards. Right? I don't have a calculator here to do that right here, and I just showed you that number because people ask me what the number is. I'm not sure I understand your question.
Well, why is it 1? I don't understand why you're multiplying. Why is it double the number?
I'm not asking you the number. I'm asking you is it going forwards or backwards? Okay. All right, so we agree it goes forwards, right? All right. Does that make sense? Okay.
Why is there a negative in the RT?
Well, don't these two have to add up to 0? Don't this and this have to add up to 0? So doesn't one have to be the negative of the other? Right?
If you rearrange it algebraically,
Yeah?
the
Yeah, algebraic rearrangement gives you that, right? Right? Question over here. Any more on these before we jump to something else? Any more on this?
My question was about the Quarry cycle.
Okay, the Quarry cycle.
I was just trying to clarify. You said it only runs when there's lactate, which is when there's low oxygen in the muscles.
Uh-huh.
But the liver is not limited by oxygen.
Okay, so our question has to do with the Quarry cycle, and I said that the Quarry cycle requires lactate in order to run, and that happens when we have low oxygen levels. And she's confused because I also said that the liver doesn't have low oxygen levels, and that's correct. If the liver gets into low oxygen levels, you're in trouble. It's the muscles that have the low oxygen, and that's why they're making lactate. And that's why they're getting rid of it, because they don't want that accumulating there. They send it to the liver where the liver has plenty of oxygen to deal with that lactate and make it ultimately into glucose. Does that make sense? So you don't want your liver to be out of oxygen. If your liver's out of oxygen, like I said, you're in trouble. I stole your pen, didn't I? Here, sorry.
So,
Yeah.
I think in the notes I saw proteoglycans are the anionic
Yeah.
carbohydrates.
Proteoglycans are polyanionic. They're carbohydrates linked to proteins.
I think they're called mucoglycans somewhere else.
They can also be called mucoglycans. You see different names for them. That's correct.
Okay.
Yep. They're called mucopolysaccharides. They're called mucoglycans. They're called several names. Yeah. And the muco tells you something about where they're found. Mucus. Let's see. Let's turn that off. Go back to our screen. Yes?
As far as elongation factors go, do we have to know more than the fact that it uses GTP to link?
Well, as far as elongation factors, what I would say is you should know whatever I said about them. Yeah, I mean, EFG had a certain function, EFTU had a certain function, and I think you should know what those functions are, yeah. Yeah?
Can you just run through plasmids and blue-white screening?
Yeah, sure. Plasmids and blue-white screening, okay? And by the way, I finally did get that figure fixed that I had a broken figure on before, so I apologize it took me a while to get that. Blue-white screening, right there, okay? All right, so when I described blue-white screening, I was describing a very simple way for us to tell not only if cells got a plasmid, but also if they got a plasmid that had an insert in a place in them. Why is that important? Okay, so imagine I've got a plasmid, I cut it with EcoR1, and I want to put an EcoR1 fragment from somewhere else in there. If I take and I mix the plasmid and I mix the fragment and I add DNA ligase, a pretty small percentage of those plasmids will find that fragment and link to it. Much more commonly, the plasmid will find its own ends, and it'll get stuck to its own ends, and there will be no insert. Well, when that happens, I don't want those, because I want something that's got the insert of what I'm trying to put into it. Human growth hormone gene, for example. Okay? So the blue-white screening gives us a very simple way to identify which of the transformed bacteria have an insert. Yeah?
Some bacteria do not have plasmids, right?
Bacteria.
That's like the first step.
So I start with a bacterium that does not have a plasmid. So I'm putting this into bacteria that don't have a plasmid. I've taken a plasmid from somewhere else, and I'm now going to put it into a bacterium that doesn't have one.
So that antibiotic resistant thing that happens that you do is looking for the ones with the plasmid.
Right, so let me set that up.
Right.
Okay, all right. So I've got bacteria sitting over here, and I've got a plasmid sitting over here. So I got this mixture, right? The mixture has got, they've all been ligated together. A few of them have an insert. A lot of them have no insert. They just ligate it back on themselves, right? Well, the plasmid had several features about it that would enable me, first of all, to tell me which bacteria got a plasmid when I try to put a plasmid into bacteria, because that also is a low efficiency. I said 1 in 10,000 bacteria might get a plasmid. How was it I identified which bacteria got a plasmid? Antibiotic resistance carried in the plasmid. So any bacteria that grow in the presence of the antibiotic that has the gene on that plasmid, I know if they grow, they had to have gotten a plasmid. Now I want to know which of those ones that grow have an insert, right? Well, I use the fact that when I put the plasmid in, I put it into the LAC-Z gene. It's in the plasmid. The plasmid's got the LAC-Z gene in it. The LAC-Z gene is the gene that breaks down X-gal and makes a blue color. So if I take this plasmid and I cut it in the middle of the LAC-Z gene and it comes back together, it's going to have a perfectly functional LAC-Z gene. And if I put this plasmid together and it's got an insert in it, my teeth, okay, it's going to interrupt the LAC-Z gene, and the LAC-Z gene is not going to work, and it's not going to be able to break down X-gal and give blue color. It's going to give a white color. No inserts will be blue. Those that have inserts will be white, because the LAC-Z gene won't be functional.
So would the restriction site be within the LAC-Z gene?
The restriction site is in the LAC-Z gene. That's correct. Yep. It's a cool trick.
So the LAC-Z gene is still there, it's just not functional?
It's not functional because it's got something stuck in the middle of it. That's right. Yes, sir.
So surprising the antibiotic is telling you that what you wanted actually went in,
It tells you that a plasmid went in. That's correct.
and then the color is telling you where.
The color tells me if the thing that went in had a fragment inserted in it or not. Right? Because I have two possibilities. When I cut that plasmid, it can come back on itself, in which case there's no insert, or it comes back with an insert. Right? I want the ones with the insert. Because that's the fragment I'm trying to put into this plasmid to grow. Okay? Make sense?
So Grow S and Grow L are type of chaperone?
Grow S and Grow L are proteins involved in functioning as chaperones. That's correct. Yep. How are you guys feeling about this exam?
Really bad.
Really bad, really horrible. You guys did this last time, and you made exactly the same average as you made on the first exam. This is good. Hard work is good. No, I tell you, I was very pleased with your performance on the last exam. I was very, very pleased with that. I think it's cool because a lot of times when I see people a class get off to a good start, they usually fall, and then they have to go back and, oh my God, I got to study, and they go back and study and they do well. But you guys did nice on the first exam, you did nice on the second exam. So I hope that you don't. I want to see you do well on this exam. I will be delighted. I will tell you what, if you guys make a better average on this exam, I have done this before with a class, we'll have a happy hour at the end of the term. How's that? Just extra credit points. But you guys already have the good scores. You won't need the extra credit points, right? You'll need the happy hour is what you're going to need. What's that? A request.
Maybe you could just change the last test to not a comprehensive test.
I would have to take away the note card.
Deal.
I don't think I would do that, but let me think about that. I'll consider that. How's that? Yes, sir.
So, concerning PCR, how do we target the gene that we are trying to replicate?
So question, you're going to do PCR. How do you target the gene that you're going to replicate? Okay? That's done with the primers. So the primers are complementary to regions around the gene you want to replicate. So you design a primer, and you have to know the sequence around the gene you want to amplify to do that.
Basically, need to know select a promoter region, and how do you select where it's going to terminate?
Okay, so let me answer that to you separately.
Okay.
I'd have to sit down with a pencil and paper and show you that. You don't need to know it for this exam, but it turns out that the termination happens when it gets to the other primer, and it can't elongate any further than that, and it falls off. But I'll show you that. Okay. I actually have a patent on PCR. Not on PCR itself, because if it were, I wouldn't be standing here in front of you teaching, but PCR involves 2 primers. I invented a process that does it with 1. And it's called boomerang DNA amplification. Yeah. Yeah. I'll give it away. If you want it, it's yours. I do have a patent on it, though. Yeah?
So the structures that you want us to memorize are glucose, fructose, and ribose, correct?
Galactose. Yes. And mannose and xylulose and what was the other one? Sorbose, yeah.
You want us to know this as well?
No. But it sure got awful quiet in here.
Do we need to be able to draw those in both Fisher and?
You should be able to draw those in Fisher and Hayworth, yes.
Just glucose and ribose? And galactose?
And fructose, yeah.
Are you going to differentiate whether to draw them in Pyrenees or Pyrenees form?
You only need to draw them in the forms I've asked you. So in the case of galactose, glucose, they're both in pyranose. In the case of ribose and fructose, they're both in furanose. And that's the ways I've given them to you also. Yeah? Ribose, glucose, galactose, and fructose. And that's the 4 most common sugars you will encounter in any of your classes. Is that what you're after also? Question? The function of the MHC complex. So MHC complex stands for the major histocompatibility complex. And its function is basically to hold peptides that show what it's been exposed to. That's the basic function of the complex. Okay? And I didn't say much about it. There are two different complexes. We don't need to worry about that for this class. But that's the basic function of the major histocompatibility class. I went through that immune system lecture pretty quickly, I will tell you. And I can assure you that I don't want you to know it in any more depth than what I talked about. That's, like I said, immunology is something that could have been done in a whole course. And so it's pretty hard to go through it very briefly and do much with it. I enjoy teaching this class, but one of the problems with the class is because we try to cover all biochemistry in one term, we can't spend much time on anything, as I'm sure you've noticed. And so we go through it pretty quickly. My favorite parts are what we're doing right now, though. I love the metabolism. Metabolism is pretty cool. Yes?
For ROS, do we need to know the specific base pair or specific?
I didn't give you the specific base pair. So the question was, do you need to know the specific? You talking about where the mutation occurs? Yeah. So I didn't give you the base pair, no. But I can tell you it affects the 11th and 12th amino acid. Would that be of help? What's that? No.
We just need to know that eco-rib is used to explain sticky.
I'm sorry, I can't understand.
We just need to know that eco-rib is used to explain sticky ends.
So EcoR, it's called EcoR1, is a restriction enzyme, and it was used as an example of one that leaves sticky ends. That's correct.
Okay.
Yeah. Excuse me. Yes, Lisa.
Cellulose, a disaccharide?
Cellulose is a polysaccharide. Oh, you're talking about cellobiose. Cellobiose is a disaccharide. That's correct.
We can't break that down to beta.
Yeah, we can't break down cellobiose either. That's right. It just occurred to me, I wonder if cellobiose is sweet. Thinking of artificial sweeteners. I mean, it's got glucose in it, right? You might expect it might be sweet. Never thought about that before. There's your $1 million idea, guys. There are some schemes I've seen where people use, I think it's L-glucose as an artificial sweetener because it has some sweet character, apparently, and you don't break it down. So.
It has to be in the lead form to break down?
Yep. And you know, when you look at stereochemistry and some of the cool things with stereochemistry, I wish I could think of the example, but there's a molecule that it has two identical structures, and they only differ in whether the D or L. And in the D form, it's a molecule that's sweet, and in the L, it's bitter. Pretty cool.
Like hops.
Hops that way?
Something like that. One is aromatic and the other is bitter.
Ah, okay. All right. Any more questions? Either I wore you out or you wore me out. Yes, sir.
What is the
Okay. What's in the rumen? Is that what you said? Yeah. So what's in the rumen is they have, ruminants have a bacterium that is actually a strict anaerobe that grows only in the environment of the rumen, and it contains this cellulase enzyme. And without that bacterium, a cow couldn't break down cellulose either. But the environment of the rumen is pretty unusual, and it facilitates the growth of this bacterium. What is the rumen? Oh, the rumen is a specialized part of their stomach. I'm sorry, yeah. I didn't understand the question, yeah. Lisa?
So besides the four that you mentioned that were different, gluconeogenesis, exact opposite of glycolysis, meaning the same enzymes are used.
So her question is, except for the 3 or 4 enzymes that are different, glycolysis and gluconeogenesis uses the same enzymes, just different opposite directions of the reaction. That's right.
And the ones that use ATP and glycolysis also use ATP and gluconeogenesis.
So no. So her question was, if they use ATP and glycolysis, do they produce ATP and gluconeogenesis? And no, they don't. No. So let's remember what we talked about yesterday. So, okay. So let's. All right. So. Okay, so there's the only places where ATP is used. And I said that we bypass those by going that way. Okay, so down here, we're producing ATP. Up here, we're using ATP. Right?
Yeah, going from 3.
Right, but this is producing ATP, not using ATP. So in the downwards direction, it's producing ATP. In the upwards direction, it's using it. I'm sorry, right here. Sorry, this one. I'm sorry, this one, right? Going down, it's making ATP. Going up, it's. Did I answer your question? Okay. I was incoherent, but I answered her question. Yes, sir.
RAS is the only proto-oncogene that you mentioned any details.
Question is RAS the only proto-oncogene to mention any detail? P53. That's a proto-oncogene. Yes.
So for gluconeogenesis, we use 4 ATP and 2 GTP.
That's correct.
Per pyruvate, or is that for 2 pyruvate to make 1?
So our question is, how many ATPs to go from 2 pyruvates to 1 glucose? So to go from 2 pyruvates to 1 glucose, it takes 4 ATPs and 2 GTPs. A total of 6 triphosphates. 6 ATPs per glucose is the needed energy.
There's a stuff that uses hydrogenase or dehydrogenase, and that just uses the energy of oxidation. Does that mean it's oxidative phosphorylation?
Oh, it's a very good question. So you've heard of oxidative phosphorylation, and here's an oxidation and phosphorylation. Does that mean it's an oxidative phosphorylation? No. So and that's why I said the oxidation is occurring in one step and the phosphorylation is occurring as a result of the energy from that. Oxidative phosphorylation actually occurs in the mitochondria. And we'll talk about that very soon. Yeah, you'll see the difference when we get to that. But I would see why you would think that. Yes?
So the 3 places that all this occurs is
Okay, so
cytoplasm, or is it cytosol?
cytoplasm, cytosol, same thing.
Okay. And then
So the very first reaction's in the mitochondria. Cytoplasm all the way up to here, and then over here, it's in the endoplasmic reticulum. But for glycolysis, everything's in the cytoplasm. Where the ATPs are? Okay. So remember, we've got to have 2 pyruvates. So it's going to take 2 ATPs here. That's going to take 2 GTPs here. And that's going to take 2 more ATPs right there. Right. So that's the 4 ATPs and 2 GTPs that's necessary. Make sense? Everybody's waiting for somebody else to ask a question. I think when you get to that point, you're pretty much done. So take care, guys. See you tomorrow. Hello.
Because I've not gotten back my first textbook.
Oh, okay. I've
Go
got
to your
it.
office, or are you leaving?
I'm going to my office if you want to follow me back.
Sure.
Okay. Yeah, there you go. I didn't realize I was as